Hi, my name is Sandra Ryeom, and I am the Program Chair of the Cancer Biology Graduate Group. And today I'd like to share with you research going in our program. We are one of the programs under the CAMB umbrella. And we have about approximately 70 different faculty who are affiliated with our program. And these faculty span departments as well as different schools at Penn. The research in the program is organized around 5 broad topic areas that align closely with the NCI Cancer Division. First, we have tumor immunology and inflammation. We also have a DNA damage, genome integrity, and chromosome elaboration group that includes this as well. There is a computational bioinformatics and therapeutics group of faculty. And then we also have a tumor metastasis group. And this includes organ-specific cancers as well as RNA biology. And then finally, we have faculty interested in the tumor biology and microenvironment. And this includes stress responses, stem cells, metabolism, and signaling. And what I'd like to do is highlight a number of different faculty in the program and briefly their research programs with you so you have an understanding of the varied research going on in our program. So first, Dr. Craig Bastian. Dr. Bastian's lab is interested in understanding how the host generates adaptive immunity against pathogens and cancer while suppressing autoimmunity and lymphoid malignancies. Research projects in his lab revolve around investigating gene expression and recombination as well as DNA repair. There are projects that are also investigating antigen receptor gene recombination and expression. And finally, he's also looking at DNA damage response and the regulation of lymphocyte development selection during development. There's a list of his current graduate students here on the slide, and all of these labs that I'm talking about, their email and websites will be available to anyone interested. Next, I'd like to highlight work by Dr. Sharon Discon's lab. Dr. Discon has an interdisciplinary lab that utilizes large-scale multi-omics data and couples this with rigorous quantitative and experimental approaches to address key questions about childhood cancers. A main focus of her lab is neuroblastoma, where the survival is quite low for high-risk disease, and currently relapse is incurable. Next, we have Dr. Karen Eisenberg's lab. Dr. Eisenberg's research is focused on sarcomas, which are a heterogeneous group of cancers that are derived from mesenchymal tissues. Here on the bottom left, you can see a schematic of all the different types of sarcoma subtypes, as well as the incidence and the survival. She is particularly interested in the tumor microenvironment and how sarcoma or tumor cells crosstalk with extracellular matrix, immune cells, and endothelial cells, and how this crosstalk regulates primary sarcoma growth or sarcomagenesis, as well as metastatic progression. Dr. Roger Greenberg's lab is focused on understanding how the DNA damage response signals at the cell autonomous and organismal level. Dr. Greenberg has 3 broad areas of study in his lab. The first is looking at the breast cancer susceptibility gene, or BRCA1 and 2, and how BRCA is involved in DNA repair. Next, he's interested in understanding alternative telomere maintenance, or ALT, and how this is regulated both in normal cells and in tumor cells. And then finally, he's investigating the DNA damage response and how this regulates immune responses, and in particular immune checkpoint blockade in cancer. And as you can see here, he cites publications from each of these areas of research from his lab. Here we have work highlighting research in Dr. Thomas Tinkanenko's lab. Dr. Thomas Tinkanenko has 3 broad areas of study in his lab. The first is to of altered splicing of cell surface antigens in tumor cells and normal cells, and how this differential splicing is regulated and maintained. He's also interested in understanding how aberrant splicing affects immune response, immune checkpoint blockade in tumor cells. And then finally, he's elucidating the role of aberrant splicing events and how this contributes to chemo resistance. His lab is also investigating whether targeting alternative splicing mechanisms can restore chemosensitivity by normalizing these splicing patterns. Here is work highlighting the research here's a slide highlighting the research in Dr. Katie Welland's lab. Dr. Welland is investigating how nutrient signaling in metabolism in cancer are regulated and their effects on tumorigenesis. In particular, projects in her lab are focused on understanding how metabolites are sensed at the cellular level and then how the cell adapts to these metabolites. She is also interested in understanding the bidirectional relationship between cellular metabolism and epigenetics and how these metabolic pathways and epigenetic pathways impact tumor growth and metastasis. And finally, she's very interested in understanding how diet and metabolism affect both tumor growth as well as therapeutic responses. And finally, I would like to highlight work in Dr. Xiaoling Yang's lab. Dr. Yang's lab is interested in understanding the molecular and cellular mechanisms that protect against cancer and neurodegeneration. He has two broad areas of research in his lab. The first is suppression from the angle of p53 regulation. p53 is a tumor suppressor, and his lab is working to understand up and downstream relation of the p53 signaling axis. He is also interested in understanding metabolism in cancer cells and stem cells. Another broad area of research in his lab is neuroprotection, and in particular, understanding protein quality control systems, as well as identifying gene therapies to target and prevent degenerative diseases. As you can see here on the bottom, a selected list of publications from his lab encompassing all these areas of research. And then finally, a Google Doc that I'm happy to send out the link to anyone interested. And what this Google Doc has listed on it is specific rotation projects available in faculty's lab in the cancer biology program. So what you'll see on the doc is the PI name, the location of the lab, the department they're affiliated, as well as specific rotation projects listed. You'll also see contact information with email and website, and there's a column listing whether or not lab rotations would be in person this fall. Anyone can send you the link. Any other questions, please feel free to email me, and thank you for your time.